Good evening, ladies and gentlemen. I'm Gerhard Kasper, and I'm the president of the American Academy. And my main task tonight is to welcome you all to what will be a fascinating talk. And after that, I should sit down because the real task of introducing the speaker will fall to somebody else. We have developed this absolutely wonderful custom that one of our fellows who gives talk should be introduced by somebody from Berlin. And we are following this this long-established tradition of just a few weeks. We are following that tonight. If lawyers know one thing, I'm a lawyer by background, if lawyers know one thing to do, they know to establish traditions in no time. And that is what we are doing here. Now, so my real task tonight is, in addition to welcoming you, is to introduce the person who will introduce Monica. And that is very difficult for me to do because Lorraine Daston, or Rainey as she is known to her friends, actually welcomed your predecessor, the predecessors of the present generation of fellows, in January. And it was at that point that I was being introduced to Berlin, and I had to introduce Rainey as the person to speak at that point. Now, you can imagine I worked very hard at it. Well, I have known Rainey for a long time, but I wanted to impress her, and I wanted to impress the rest of Berlin. And so I did one of my more elaborate introductions. And when I say one of my more elaborate introductions, that says quite elaborate. And it's a wonderful introduction. You can go to our website. You can go to our website and listen to it there. It's on video, live, kind of, as it were. And I hope you will, of course. But in any event, I will not do that. No. I will just very modestly say something that I said that evening, repeat that, just one sentence. No, it's three sentences, but they are very short. I said it should be sufficient to say that Lorraine is one of the most distinguished historians of science in the world. And historian of science, I went on to say, does not quite capture it. She is interdisciplinarity incarnate. And that is my three sentences. But then I went on to quote one of my Stanford colleagues, and he should be quoted again tonight, Jim Sheehan, who said about Rainey, Daston's scholarship reflects a remarkable range of interests and erudition extending across chronological and national boundaries, including evidence from art and literature, philosophy and theology, as well as scientific theory and practice. I give you Rainey.
Thank you, Gerhard. I'm really looking forward to the third round of this in which you have condensed it to a haiku. So in the spirit of newly minted traditions, as my accent betrays, I am hardly a geborene Berlinerin, so you will have to take introduced by someone from Berlin, cum grano salis. I was trying to remember the last time, or the first time, that Monica and I had seen one another. And I think it was in 1984 when I arrived in Princeton as a wet behind the ears assistant professor, and Monica was finishing a dissertation on the transmission of knowledge about female physiology in the Middle Ages. So when I got the American Academy's roster of fellows and topics, and I saw the name Monica Green and the topic, I thought, oh, it must be another Monica Green. There must be a historical epidemiologist. And about 2 weeks ago, my error was set straight, and I realized that the scholar whose work on Trotschiller, which I have been following with keen interest and admiration for so many years, has many other arrows in her quiver. From what I've just said, it should be clear that Monica Green is a historian of extraordinary breadth in every sense of the word. Geographically, her work has encompassed the vast expanse of the medieval Norman holdings from southern Italy to England, and indeed wherever medical knowledge circulated, from Muslim communities in northern Africa and the Iberian Peninsula to Jewish communities in Italy and the Holy Roman Empire to Christian communities, both Eastern and Western. More recently, she has ranged still further to the Tibetan Qinghai Plateau and along the trade routes of the medieval globe, from the Silk Road to the Indian Ocean. Chronologically, her range has extended in both directions from her home territory of the Middle Ages. She's gone back centuries to the reign of the Roman Emperor Justinian, and she's gone forward to the most recent outbreak of the Ebola virus. Thematically, she has written on the history of women's health care in the Middle Ages, the spread of both learned and vernacular medical knowledge from the famous so-called School of Salerno, relations among Muslim, Christian, and Jewish women in the High Middle Ages, the history of the body, and the reconstruction of diseases such as leprosy, the bubonic plague, and other long-lived afflictions, both on the basis of historical records and paleogenetic records. I should also say, and this was clear to me when I first met Monica, Monica is a historian of extraordinary courage. When I first met her, as I said, in the early 1980s, her dissertation topic, which now sounds not only perfectly plausible, but actually quite mainstream, on the history of ancient theories and of female physiology and their transmission in the early Middle Ages, if that's the case, it is only because of the vision and bravery of pioneering medievalists and gender historians like Monica, Joan Cadden, and Catherine Park, who showed how knowledge about birth, sex, sexuality, that is the common preoccupation of all peoples everywhere and always, circulated across barriers that separated lay from learned cultures, rich and poor, men and women, Muslim, Jew, and Christian in otherwise highly stratified societies, and circulated also across continents and centuries. Thanks to the efforts of Monica and her colleagues, we now know that this medical tradition is among the most long-lived, widespread, and influential of any intellectual lineage anywhere. But no one who was not at Princeton in around 1980, and some of you were, I think, can imagine the outright sneers and incredulity which such claims then encountered, and how much determination and just plain moxie it took for a graduate student, nonetheless, to persevere. It's also taken considerable courage to campaign for the relevance, indeed the indispensability, of integrating the latest results of paleogenetics, that is, analyzing the DNA of organisms that died centuries or perhaps even millennia ago, to integrate that with standard historical narratives. The reflexive reaction of historians to such new evidence has, alas, been largely defensive. They have feared a lapse into the most flat-footed kind of positivism, the sort that accumulates facts without knowing what to do with them. And I must say, I have seen examples of this sort of fact-mongering, ohne Fragestellung, as they say in these parts. And it's no secret that humanists, both in North America and in Europe, find themselves increasingly battling to justify their very existence, and they are weary of having the apparently more practically useful sciences held up to them as a standing reproach. In the teeth of such disciplinary skepticism, Monica has seized upon the potential of this new evidence to deepen the narratives historians have already constructed on the basis of textual and archaeological evidence about episodes like the outbreak of the Black Death in Europe of 1348, which eventually killed somewhere between 40 to 60% of those it attacked in urban areas. Moreover, she's forcefully argued for the necessity of a collaboration of equals between historians and geneticists to weave together causal strands, natural and cultural, that create ecologies in which bacilli, like the Yersinia pestis, do and do not flourish, can and cannot be transmitted. In the age of SARS and Ebola, I hardly need belabor the urgent relevance of such intricate causal accounts for the here and now. Together with her collaborators, Monica is not only blurring the line between the humanities and the sciences, she's also blurring the line between those long-drawn categories, the natural and the cultural. Do these distinctions really make sense in the context of historical epidemiology, in which these causes are constantly interacting in ways too intricate to disentangle? Monica's achievements have been honored with numerous prizes and fellowships, including the Margaret Rossiter Prize of the History of Science Society for her book, Making Women's Medicine Masculine, The Rise of Male Authority in Pre-Modern Gynecology of 2014, as well as fellowships at the Princeton Institute for the Advanced Study and the Radcliffe Institute at Harvard University. Since 2001, she has been professor of history at Arizona State University and has held visiting professorships in Utrecht and Seattle. Her trailblazing work is helping us to rethink the meaning and history of the global, as well as the always precarious, always complicated, always contested preconditions for health. We look forward to hearing Monica's latest thoughts on these topics in tonight's lecture, and she has asked me to announce that we don't really have enough handouts for everyone, so please share. Please join me in welcoming Monica Green.
Thank you. Just one correction is that I deliberately didn't make enough handouts so that you would all share and we would foster more interdisciplinary dialogue by people contesting over what's in the handout. So please do share. Thank you, Rainey. Thank you, Gerhard. Thank you, everyone, for coming this evening. I'd just like to start by I have acknowledgments at the end, but I'd like to dedicate this talk to my mother, who passed away last year. She was of Irish-German background, and when I was in 7th grade, I actually did a little research project on one of the German ancestors of our family, and I had my sister dig up this report. And the name that I wrote down, remember I was in 7th grade then, I cannot find anywhere in any record of a real German town. So I can't really tell you where we're from, other than it is said to be some part of Bavaria. The other part of my family history, I just want to mention as a way to get us thinking about global issues. My father was African American. And, a few years before he died, he had his genetic ancestry tested by one of these commercial companies. And it said that he came from Cameroon and from Niger. And so that's part of where I come from, is this global mix of people. And that's part of what I want us to think about today. Okay, so in my 3-minute talk last month, I gave you 3 numbers. I gave you 8, 13.9 million, and 1. 8 was for the number of diseases that I'll be incorporated into this project that I'm working on this semester. 13.9 million, as I'll explain soon, is the estimated divergence point of the 2 types of leprosy that have been discovered. 1 is the number of transmissions it took to pass what was the simian immunodeficiency virus into a human that became the HIV pandemic that we are still living with today. Except for leprosy, I won't be talking about all of these diseases today, but I want to give you one more number, which I'll get to later, but I just want you to think about it right now. 1/3. Okay, so what I want to do today is give you a sense of what the whole project is, rather than just a section of it. And I want to say that I don't think it's bravery, I think it's necessity to think in these very broad terms and think across disciplines, because I think that we have a lot of very, very urgent and very big questions that we need to wrestle with as a species. I want to give you some sense of the scope of the project, the methodology or methodologies that I'll be using, and then also the goals of the project. So the handout that you have, and if you didn't get a handout and you really want to have a handout, I have this same file posted on my academia.edu page, so you can find it and download it there. This is a summary of all the diseases I'll be talking about in the book, and I won't be going through all of anything of this tonight. But these are some of the highlights that I want us to think about of the kinds of knowledge that we have, the kinds of knowledge that we can have of these diseases. So just to point out some of the things that I'll be talking about. So the disease, the common disease name, the causative organism, earliest impact on humans. That's the main thing that I want to talk about. And just want, as a conceptual issue I want to make sure everybody understands at the outset is I'm only focusing on infectious diseases. Why? Infectious diseases means that it's something that comes into the human body and infects. But it also means that it's a something, whether it's a virus, whether it's a bacterium, whether it's a protozoan, that does exist, live, evolve outside of human bodies. So they actually have separate histories. And that is the great beauty of why this project is possible and why it has the scope that it does. So infectious organisms, so causative organism is going to be vital. Earliest impact on humans, so when is the earliest we can say this organism was getting into human bodies and causing disease? Discovery of causative organism, this is the old history. This is the late 19th century history of laboratory medicine, much of which happened right here in Berlin. Genome sequenced, you will be hearing a lot about genomics today. But if you'll just run down and look at the dates there, smallpox, 1994, and HIV/AIDS in 1985, those were the front runners of genetics research. Everything else has been much more recently. Development of effective treatment. I won't be going into this aspect of history of medicine at all, but I do want you to note the final column there, documented drug resistance. That means whatever mechanisms we have developed to deal with these infectious diseases, with the one exception of smallpox, are being compromised in some way, that the organisms are fighting back in some way and resisting the drugs that we have developed. Why is smallpox the exception? One, we eradicated it. Two, we never had an effective therapy for it in the first place. We eradicated it only because we had a vaccine. Okay, so I won't be going back to this. You can refer to the handout. And please, I will take any kind of question at the end. So anything that you want me to go back to revisit, please go ahead and raise the question. Okay, so scope of the project. Why am I going global? Because humans have gone global. And all of these 8 diseases that I am looking at have gone global. All of them. So there's, even though everyone started somewhere as a local outbreak, and maybe even for centuries was a local disease, all of them have been distributed globally. In terms of time, I also want to think about the entire history of Homo sapiens. And why is that? Is in part because some of these disease histories, in fact, go back that far. Some of them perhaps even go further. But part of the story that we need to think about is human migrations. So HIV is globally distributed in part because of airplanes. But airplanes are only a really central part of the globalization of just that one disease. All of the other diseases had been globalized long before airplanes were around. So it's humans that have taken most of these diseases where they are. A few exceptions, but it's mostly human migration. So this is just a basic narrative. and which I'm always having to change this map because there's always new studies coming out. So that was a map from 2007. This was a study that came out in 2011, which was adding the new Denisovan DNA and suggesting the path of the colonization of Australia. And this just came out last week. I can't even go away for a weekend without these new studies coming out. So this was a new study that is pushing back several tens of thousands of years to as much as 120,000 years ago. The first migrations of Homo sapiens out of Africa and into the Eurasian continent. I try to keep track of these permutations as they come about. In fact, there are a lot of the dating issues that are, for a historian, astoundingly flexible that one has to keep in mind. But this is just part of what I want you to keep in mind is the origins of Homo sapiens in Africa and migrations. And those maps don't capture the layers of migration upon migration upon migration, which will bring us up to the present day. Okay, so thinking about scope in space, thinking about scope in time. I will talk more about my use of genetics, but this is just an example of why we need to think in terms of very, very deep time. Because it changes the parameters of the phenomena that we think that we're dealing with. So the number I gave you, 13.9 million, is the point at which right there in the middle of, I have a pointer, disregard that 100. But right here is the point, it would be called the most recent common ancestor of M. leprae and M. lepromatosis. Those are the two bacilli that now are understood to cause leprosy in humans. And that point of divergence is now estimated on genetic grounds as 13.9 million years ago. Humans did not exist 13.9 million years ago. We had not yet separated from chimpanzees. So this is whatever is happening to cause this split between the two forms of leprosy. We as humans didn't do it. But then the question is, where do humans come in to this story if this organism has separated that far back in time? Now, the other thing is global distribution. The M. leprae that most of us think of as the leprosy most of us think of and is globally distributed is M. leprae, which was discovered in 1873 as an organism by the Norwegian researcher Hansen. M. lepromatosis, for the most part, is currently confined to Central America. But if they both started in the same place, how did one become globally distributed and one get centered just in one part of the world? I don't have an answer to that, by the way. But this is the more important. So here is that same, we call this a phylogenetic tree, of showing the evolutionary development. So there's M. lepromatosis, M. leprae. And then that one bar there is here expanded to the whole history, as we now understand it, of M. leprae. So 13.9 million years, this organism has been in existence, and it's replicated. I mean, that's the thing about these are living organisms, so they have to keep replicating for them to still exist in the world today. The divergence time for the most recent common ancestor for all the M. leprae strains is only about 3,607 years. This is a computational fiction, if you will, but let's say between about 2,000 and 5,000 years ago. All of the variation that we see in leprosy in the world today happened only in 5,000 years. Is 5,000 years ago the first time that humans, hominids, were infected with leprosy? We have no idea at this point because we don't know of any other animal reservoir for it. So this is just an example of how we need to think big, how we need to think globally, and even then, sometimes we don't get the answers that we want, but we know that there are big questions there. Okay, how old is malaria? I'll just go through this quickly. This would be an hour lecture in and of itself. This is all genetics work that has put together this story. The two main kinds of malaria that afflict humans are vivax, which is very broadly spread, and falciparum, which is mostly confined to Africa and a few other places. Just the color coding here can give you an idea, if you're looking at the relationship here, that humans with the Plasmodium falciparum got that kind of malaria from gorillas, this falciparum. And the vivax that we have now was widely distributed among various primates in Africa. Vivax is the older form of malaria, and really a fascinating history about essentially vivax being more or less driven out of Africa, and then allowing space for falciparum to thrive and create one of the deadliest diseases that we have in the world today. So anyway, this is just a sense of why we need to push back in time. And there's no time indicated on this. I would love to know, but we don't have any ADNA for malaria. And I'll explain in a minute what ADNA is. Okay, so what I am doing as a historian, I am broaching into these other disciplines, genetics and bioarchaeology, paleopathology, to get their goods, to say, you can give me historical information, I want it. So as a historian, I was trying to work with documentary sources and still do that. Works of art we can also bring into our analysis. But this is the work of other fields, and so I'm a shameless poacher in taking advantage of this other work in bioarchaeology, paleopathology, paleoepidemiology, and genetics. So ADNA, as I use it in this lecture, just means ancient DNA. DNA fragments that have been recovered from organisms of the past. Okay. Basically, what I'm trying to say is we have to get along because none of us has all the goods. No one discipline produces all the kinds of information that we need to put together a rich and coherent interpretation about the history of these major human diseases of humankind. So what was the disease? Just thinking about that sheet of just saying, is it Mycobacterium, leprae, or some other organism? Genetics is great at that. Paleopathology is often very good and in some cases quite persuasive. How old is the disease? Genetics is still, I think, a little shaky. Paleopathology and history. The SO there stands for this highly scientific concept that I came up with. I mean, sort of. So, yeah, we can fudge and we can make things up, but not always reliable. Where did it come from? Putting the geography into the story. Again, the genetics can sometimes give us some very decisive information. Sometimes it's just hints. Paleopathology is precise. They're digging up the bones. And so wherever those bones are, that's where the disease was. History, how many were afflicted? Asking the epidemiological questions, genetics is well, almost worthless. So there's certain kinds of population genetics that can be used for this, but it hasn't been done so much. Paleopathology, depending on how good the samples are, how big the cemeteries are, et cetera, sometimes they can do it. History, okay, how did people respond? That's what we're excellent at as historians because we are searching for humans. We're not looking just for the microbes or other remains. Okay, so paleopathology. So history, I will just assume that you have a sense of what historians do. Paleopathology is dealing with the physical remains of disease that has left its imprint on human bodies. Having mummies is a rarity. Having mummies this finely preserved, where it has been said that the lesions on the skin that you can see there of Ramses V were caused by smallpox. No genetic material confirming smallpox has been recovered. There are some possible virological traces. But that's one of the things that has traditionally been used is just looking at these bodies. But paleopathology actually can do something more. Now, what paleopathology does is work with mummies when mummies are available, but mummies are rare. Bones and teeth are the hard parts of the body that usually survive best. And this is just fairly recent, is figuring out that smallpox, even though we don't have smallpox in the world anymore, realizing that there might be distinctive traces that can be found in the skeleton to show someone who has survived a smallpox infection. So this is a study from 1998 in smallpox epidemics. 2 to 5% of children and adolescents develop osteomyelitis and arthritis variosa. So arthritis, variosa means smallpox, so a specific kind of arthritis. 2 to 3 weeks after the skin lesions appear. Skeletal involvement is not seen in adults, probably largely because their skeletons have already formed. So a report from Zimbabwe in 1961, when there was still active smallpox there, suggesting 20% incidence in an outbreak in Rhodesia. A rare hemorrhagic type produces heart failure, diffuse bleeding, and bone marrow depression, so that the bone marrow just stops producing new red blood cells, white blood cells, and platelets. So as a historian, this is the kind of source that we would use and has been used very frequently for the history of smallpox. So not this particular quote, which is as far as I know, is not well known. This is al-Razi, who is a Persian physician working in Baghdad and Rai in modern-day Iran. In his text, the Hawi, there was a patient who had a severe attack of a bad form of smallpox. We bled him before he felt much suffocation in his throat, and there was no cooling medicine which we did not give him. Accordingly, his throat began to be less constricted, and he was getting better from the smallpox, so that we had good hopes of his recovery. Then later, the patient was seized by a very acute pain in his leg, and the blisters turned black, and he died of the violence of the pain that same day. I had intended to scarify that part of his leg, but his strength began to sink within an hour, so that I had no hope of saving him. And at last he fell into such a state that blood began to flow from the pores of his skin. I had seen several patients in whom large blisters of a certain type came out, and who had, towards the end of the disease, a pain in the leg. So this is very typical of what we would used to call a retrospective diagnosis of the verbal description that al-Razi is giving. Sounds like it might conform to the extreme hemorrhagic form of smallpox that I've just indicated. The historians of medicine, and I won't go into this background, but historians of medicine have backed away from using historical texts for this purpose. But this is where I get excited, is that the paleopathologists have jumped into the fray and said, wait a minute, this is actually a documentable condition in survivors of seeing the ways the bones have been deformed. And again, this is the people who survive the disease have these kinds of changes. So these are from samples taken and photographs taken in the 1960s. This is an example of a way to push this back into historical time. This is just a study that just came out 2 or 3 years ago. A case of osteomyelitis variosa found in the remains from Pont-sur-Seine. Adolescent male between the ages of 15 and 17. Pont-sur-Seine is on the road to Paris. We had a talk a couple of weeks ago about the rivers of France. And this is exactly one of those cases of a riverway as being this main conduit between Burgundy and Paris. And what I think was happening, these are some photographs of this skeleton that was dug up from this grave. And it's, I think it's radiocarbon dating for getting him to the 11th or 12th century. What I think is happening, this is an interpretation that I'm giving of smallpox, is that in places like Baghdad, which, as I said, Al-Razi is, smallpox has become an endemic disease, where he's mostly seeing it in children. In a place like this, it's probably not yet endemic, and so you see it. I think you will probably see it. I think if there were more studies like this, you would see it more often, is that it's in these what I call corridors that are connecting groups of large population to others. So anyway, that's near where the skeleton was found, near Troyes. Okay, so I'm just drawing in this kind of evidence when I can find it to reinforce and sometimes to extend the work that can be found in historical sources. I won't go through all of this. This is a classic phylogenetic tree. This is the tree that was devised for plague, Yersinia pestis, in what I still think is really major study that came out in 2013. So the most recent common ancestor is where this particular species branched off, likely branched off from another one, and they're guessing about 3,000 years ago. BP in these slides, by the way, means simply before present. And then that is the point where we have ADNA, genetic material, a complete genome sequence was reconstructed from a Black Death cemetery in London. So these are people that we know died in the Black Death, buried in London, and the entire genome was resequenced from genetic material. And then that's just some other ways in which I think historians can learn how to interpret this material, because one of the things I was pointing out in a paper I did last year was, if this right there in the center is the Black Death cemetery in London, why do we have the next closest? So that's the Black Death genome right there. Up here, which as you see is the next point on that branch we get a sub-branch, that's in Africa. That's in sub-Saharan Africa, in Uganda and Democratic Republic of Congo that those samples come from. So we have some explaining to do to figure out how this disease that we used to think only had afflicted the western part of Eurasia is in fact getting to sub-Saharan Africa as well. Okay, so what I want to do now is give you a story of just one disease, which is tuberculosis, and that's where we come to our 1/3. 1/3 is the estimate that has been tossed around. There's no way to prove it, but it's an estimate of the number of people in the world today who have tuberculosis in their bodies. 1/3 of people alive today have tuberculosis in their bodies. So as a historian, how did that happen? How did that happen? Okay, tuberculosis, actually we can do a lot of things together. So tuberculosis, in certain cases, if it progresses to a certain level of severity, will leave lesions on the bones, particularly in the spine. So that's it's called kyphosis or Pott's disease, which is it's essentially the vertebrae are just collapsing, and you get a very distinctive and for the person to whom this happens, very painful hunching of the back. The psoas abscess is another distinctive thing. It's in this case, we have a mummy with some of the soft tissues surviving, where we can see the traces of this. But this is unsurprising for us that we would see traces of tuberculosis in an Egyptian mummy. Okay, again, we want to think globally. It has been recognized for decades that the same physical signs of tuberculosis infection can be found in pre-Columbian remains in the Americas, both in North America and in South America. Okay, so what is our natural assumption as historians? If tuberculosis is in the pre-Columbian Americas, then it must have come when the pre-Columbians migrated into the Americas. So whether you want to date that to 13,000, whether you want to date that to 17,000, whether you want to date that to 21,000, and then all of those things are still viable in terms of the migrations to the Americas, but many thousands of years. So tuberculosis as a disease must be older than that. And that, in fact, has been what everybody has assumed. So here's some more examples of tuberculosis from pre-Columbian samples in the New World. This is Chile. That kind of collapsing of the spinal column that I said, this one over here. I mean, one wonders what kind of pain this and dysfunction this individual had been living with. But that is very severe infection. And again, these are well dated. Mummies that have been found as well. So that's the complete mummy, and then there's an X-ray there to see the kyphosis in the spine. And that's, for those of you who know the American Southwest, that's Cocopele, which is an Indian figure very common in art, and it has been suggested that that curve in his back is a sign of Pott's disease. But anyway, we have lots of evidence. And so this is just a summary map just showing these places in on the west coast of South America and scattered in various places in North America. So these findings were made initially on the basis of just the bones, lesions in the bones. But as of 1994, there was also some fragments of aDNA that were discovered, so confirmed that, in fact, this was tuberculosis. So this was the old understanding of tuberculosis in the Americas. So tuberculosis is age. Again, it's spread throughout all human populations. When you have when you're pushing it back deep in time, that gives you a lot of time for that kind of spread. So an estimated age, some people said 35,000 years, others said 70,000 years, but many, many thousands of years. Place of origin, Africa. Its route to the Americas via Southeast Asia or East Asia, but through the Beringia transit into the Americas. The age in the Americas, again, assumed to come with the first peoples. And the evidence, which we've just looked at, is the evidence from the bones and then the aDNA. Okay, it was a nice story, a coherent story. Except it's been challenged now by this amazing work that was just published by Voss et al. And several of you, I was talking about Johannes Krause. Johannes Krause is Kristen Voss and Johannes Krause do much of their work together. So this is from that same laboratory. Okay, so they went. These are now geneticists going back to these mummies and extracting genetic material from them, able to reconstruct 3 complete genomes of the tuberculosis that was in these mummies from pre-Columbian Peru. And what did they find? They found that taking all the studies that have been done, genetics studies of modern tuberculosis, and putting it together with the complete genomes that they had reconstructed from the mummies, they said, this looks like it's only about 6,000 years old. Not simply these Peruvian mummies, but the whole history of tuberculosis in humans is only 6,000 years old. So I hope you're noticing what the problem with that is. The form of TB they were infected with has as its closest extant relative the kind of TB now found in seals. Tuberculosis is a human disease. Maybe cows, But seals? Okay, so problem, of course, is if you're narrowing, collapsing the historical focus down to 6,000 years, then you can't get migration across the Bering Strait. Or you would have to have some second crossing of new peoples that brought it over. But the whole Bering Strait crossing collapses if you're shortening the amount of time. In terms of the seals, tuberculosis has in fact already been established as being present in seals. And it's in several different seal species that inhabit the waters around Antarctica. So there are southern fur seals that are found along the Pacific western coast. Remember the TB that we saw before was right around there in Peru. But all up and down this coast we have these southern fur seals and then other species of seals that inhabit really South Africa and over into Australia. Okay, so there are two issues, and I'll explain the African one in a second. Actually, let me go back to that. Just so fuse this into your brain, is that the several species of seals are swimming around the Antarctic. And actually, if you put it back into the shape of a globe, that's a much smaller space. Okay, so we have a question of tuberculosis in seals. The seals are swimming up the western coast of South America. It is well documented that the inhabitants of those areas along the coast had been been hunting seals and incorporating the seals into their diet. So that wasn't a problem. The idea that some infection in the seals could have been transmitted to humans in South America. So, but that raises the question, that establishes the transmission to the people in these areas around Peru. The question is that same strain found in other peoples in the Americas? Because as I said, it has already been documented that tuberculosis is found in North America as well. Somebody else is going to have to research that. In fact, I'm sure research is ongoing about that question. So anyway, that's just a way in which those findings really complicate the traditional narrative. There is, however, an African question. And that was the question I immediately asked, because again, I am approaching this as a historian, and I already knew something about this strain of tuberculosis that was in seals, which I'll tell you about in a second. But then the question for me is, okay, if the seals brought tuberculosis to South America, how did the seals get it? And this is the problem. This is the problem. Okay, the purple, the blue, and the red strains of tuberculosis down on the lower half of this I'll be talking about later. But right now, we need to talk about this cluster right in here. So one strain of tuberculosis called M. africanum, West Africa, is found in humans in West Africa. Another strain, M. africanum, West Africa 2, also found in West Africa, also found in humans. But look where these animal strains are. So this includes a kind of tuberculosis that affects cows, goats. These are all quite specialized strains. In fact, voles, and several chimpanzees. In fact, it's also been found in chimpanzees. But notice this. This is, I'm sorry, I'm thrusting you into genetics analysis here, but this is how you read a phylogenetic tree here. Is you look, those are the branches. So these are related. And then this one branches into two strains. This one, because it's further to the left, means it's older. So this brown West African M. africanum is older. And then it branches into two strains, into a younger West African 2, and then to these animal strains. What does that mean? It means the animals got their tuberculosis probably from humans. So this question, M. pinnipedi is the type of tuberculosis in the seals. Pinnipeds are seals. So where did this come from? So the assumptions are, given its place on the phylogenetic tree, it must have a source related to M. africanum. M. africanum now is only documented in West Africa. All the other strains, as I said, are only, they're not simply derivative of M. africanum, but they are also only documented in Africa, except for M. bovis, which is globally distributed. And so how does this all fit together? Okay, so this is the strain of TB found in the 11th and 12th century Peruvian mummies, right in here. And of course, the explanation that Boss et al. Have given is that the seals are swimming to the coast of Peru and then giving this infection. But how did the seals get it? How did the chimpanzees get it? When this strain, this green one and this brown one, are human strains. Well, this is what I came up with. Again, I'm a historian and I'm trying to create a narrative. That makes sense. So if M. africanum is an African strain of tuberculosis, if M. africanum is a human strain of tuberculosis, then that raises the possibility that the seals got their TB from humans. So one of the claims that Boss et al. Made in their study was, while a human transfer of the bacterium to marine mammals cannot be ruled out from our data, we consider this extremely unlikely. Humans did not herd or farm seals. And close regular context would be required for anthropogenic transmission from human to animal, as is observed in domestic cattle. So it has been accepted actually for quite some time that bovine tuberculosis, the kind of tuberculosis that cows carry, was given to cows by humans. It wasn't cows that gave it to humans, it was the other way around. So we've recognized that and accepted that as a likely transmission point. But they're ruling it out for the seal strain. Well, what I did as a historian was do what I always do now, which is get on Google and look for seals Africa. And what I found were these studies by an archaeologist who works out in South Africa, evidence for both the hunting and systematic slaughter of seals, what he calls a sealing camp, at Kastelberg on the Vredenburg Peninsula northwest of Cape Town, South Africa. So here's Cape Town, there's the Vredenburg Peninsula, and this is the place, Kastelberg, where he found evidence. Exhausted, mountains of bones of these seals that had been slaughtered and taken to another camp. So the seals are slaughtered on the coast, but then taken inland to a camp about 4 kilometers away. This is a prime nesting spot for these Cape fur seals. So this is a different species. But remembering all of these seals swim in the Antarctic Ocean. So Kastelberg is used by a herding group, possibly the Kokoen, for a period of at least 1,000 years, starting in the 1st century CE. Archaeological remains give ample evidence of systematic harvesting of seals, as much for their fat as for their meat. Local herders took advantage of the seal's breeding season to target the animals while in a vulnerable position on land. Boss and his colleagues have put the evolution of M. pinnipedi, the seal tuberculosis, within the past 2,500 years, which fits with the arrival in South Africa of the seal herders nearly 2,000 years ago. So the missing link, I just put this out, this is waiting publication now, but the missing link is I don't have any proof, and I asked some archaeologists in South Africa if they had any proof of tuberculosis being in South Africa in remains 2,000 years ago. First of all, they don't have that many remains, human remains from that long ago, and certainly no TB. So this isn't a complete story. It may be proved completely false. But the point is, this is what I'm trying to do, is create consilience between the different disciplines and say, is this a plausible story? Because we have to get tuberculosis out of Africa and over to South America somehow. Okay, so bringing the tuberculosis up closer to the present day. Here's another mommy from Hungary. This is a woman, we know her name, we know when she died, we know how old she is. This is, we get really excited when we have that kind of information. Her name is Theresia Hausmann. She died in 1797. She was 28 years old. And this is what her X-ray looks like. This is as profound, the impact of tuberculosis on the spine as you can get. So this, and there were a couple of other mummies found in this tomb. This was discovered several years ago. Just this year, earlier this year, a genomic study was done. And what they found was she had 2 different kinds of tuberculosis circulating in her body. There was another one in the same crypt that had 3 different kinds of tuberculosis. This is an incredibly diseased population that is carrying this tuberculosis. It allows them to create a most recent common ancestor for the lineage going back to the late Roman period. Again, that's an estimate, but assuming a fairly constant rate of genetic change. This correlates with the SEAL study, again, saying that tuberculosis as a human disease probably has only been somewhat less than 6,000 years old. Okay, so the rest of this I will go through quickly, which is simply to say that the genetic study, the historical study, the paleopathological study are creating this coherent understanding of tuberculosis, which is actually suggesting that tuberculosis got increasingly worse in human societies over the centuries. We have incredible levels. I mean, this is just global history 101 of how much human migration is happening in the 18th, 19th, and 20th centuries. Huge numbers of people moving around and potentially carrying new strains of the disease with them. So most of the histories of tuberculosis that have been done have focused solely on Europe. And one of the things I am trying to say with this new genetic studies is that we can push beyond just written sources and take the genetics as part of our data and really make these stories global. So I want to ask these questions about why tuberculosis becomes even worse for so many indigenous populations in the course of the 19th and 20th centuries. So these are studies, figures put together by Sam Roberts for white and non-white differentials in the United States. Similar differentials, I mean, sometimes phenomenal differentials are found between whites and Native Americans, again from data in the United States. This is data that was assembled by Randy Packard for his study on tuberculosis in South Africa, who had some indication that tuberculosis preceded the arrival of Europeans, which, again, it's some very not substantial. But again, showing that blacks now in South Africa are far more heavily affected by tuberculosis than the immigrant whites. And my question is, can genetics give us some kind of answer about why we're seeing these kind of differentials? Well, remember when I said look at the red, the purple, and the blue? These are now reversed here. They're on the top of the tree here. But look at the map. Where's the red? It's not simply in Europe. It's the European strain, but it's all over the Americas. It's in various parts of Africa, it's in Asia as well. The blue strain as well, it's also called the Beijing strain in more recent studies. Those are actually have been documented clinically to be the most virulent strains of tuberculosis. So this is very much likely. It's certainly a good enough hypothesis right now until we get more evidence, is that whatever strains of tuberculosis were in other parts of the world, this European strain and then the Beijing strain swamped them. And that's why we get the distributions that we have. So this is the way in which genomics can help us create a really global history of why we have the diseases that we have in the world and why they are where they are. Okay, so this is just a summing up. I'm just going to skip through this. The point of this last section is simply to talk about tuberculosis in the world today, and the ways in which co-infection with HIV has, well, two things have happened. Co-infection with HIV has exploded because TB is actually one of the diseases HIV loves as an opportunistic disease, to take what is a quiescent, controlled infection in someone's body and then blow it up and kill them. The other thing that's happened is drug resistance. And not simply do we have extreme drug resistance, multi-drug resistance, extreme drug resistance, and now we have something that's been labeled total drug resistance, where strains of TB are proving themselves to be not controllable by any drugs that we currently have in the arsenal. So this is my argument for the presentism of my project, is that I absolutely want to argue that there is no point at which some veil dropped and we were separated. With smallpox, yes, it was eradicated. But with everything else, we are still living with these diseases, we are struggling with these diseases, and the kinds of controls that we have established for many of these diseases, we're losing. This is a history that is our present. And so I will conclude with this, which is to say that I think we watched all of this in real time in these past 2 years of the Ebola outbreak. One of the arguments that was made as late as August of 2014 is summarized in this chart. This appeared in the Economist, as saying, oh, Ebola is not a big problem. Diarrheal diseases are a big problem. Tuberculosis is a problem. Malaria is a problem. HIV is a problem. This is just data taken from the 3 West African countries that were most immediately hit. This quote, it's not from Thomas Friedman. It's the interviewer asking Thomas Friedman, the head of the CDC in the U.S. I'm trying to understand how all of us who could have done more earlier somehow missed it, and why that was. I would say one of the reasons was we didn't have this bigger history in our head. We have been going around thinking we are separated from this very long history of humans' encounters with disease. We are not. And that what we saw in the past 2 years was this process of a new disease coming into being all over again. And this is just an amazing analysis that to counter the kind of, oh, it's insignificant argument that was made almost the same day that this came out. This is a blogger, Greg Ladden, who simply said, actually, if you, instead of taking annual figures for how many people have died annually of a disease and just put it into months, in fact, by this point, this is by August of 2014, already the numbers of people dying from Ebola is matching the numbers of people dying from malaria. This is not an insignificant disease, as we learned to our great misfortune in the subsequent months. Okay, so are there ends of disease? Smallpox, we think we have an end, except we're living with bioterrorism and millions being spent on threats of these diseases. But with Ebola, through luck, through amazing determination, through astounding courage of workers in the field, and also with finally that last point was the vaccine came in. We got to this point 2 weeks ago where there was 2 weeks in a row, no new Ebola cases. this week there are 2 new Ebola cases in Guinea, and of course the relapse of the Scottish nurse, and of course, and remember this. Remember what I said about the smallpox effects. The survivors have real effects, and we should be looking at what it means to survive one of these truly catastrophic diseases. So with that happy note, I want to thank all of the people I want to thank, but I want to thank you, and very much looking forward to discussion.
Thank you very much, Monica. I understand that there is a tradition, no doubt equally fresh, that formal discussion is about 15 minutes, and then we continue for informal discussion at the reception. The floor is now open for questions and comments, and if you, since we don't all know one another, if you just say your name before you start. Philip, yes.
Thank you very much, Monica. That was fascinating. Can I ask you something about the seal story? So if the herders are being really successful and they're just running out and rounding up some seals and slaughtering them, it doesn't look as though it's going to be likely that you're going to get transmission from humans to seals. So
what if the seals turned around and ate one of the people that was trying to hunt them?
So my question is, what do you think the mechanism of transmission is from humans to seals? I mean, seals don't normally eat human beings.
I wouldn't want to mess with them. Certainly, I don't know. I have no expertise, and I pursued this question simply because I was frustrated with this study that, I mean, they have phenomenal data about all of these different strains of tuberculosis. But and literally, it only took me about 5 minutes on Google to get some raw information. Then it took me a couple more, 48 hours. It took me, because I had to write, I found a name of somebody who did this research in South Africa. I wrote to him, he wrote me right back. I mean, that's how easy it was for me to put together at least a scenario in which it might be possible. There is a bigger problem, which is something is going on, whether it's human cultural activity or some other factor that nobody has any idea about. Because the fact of the matter is we have to explain how the tuberculosis got into cows, how it got into voles, how it got into goats, so all of these other strains. We have the genomic sequence of all of these now to prove what the connection is. But the mechanism and even the timing of the connections is still unclear.
Can I make a constructive suggestion?
Certainly.
I mean, imagine these people going out and trying to get all the seals they can and injuring some. And thinking about the possibility of stowing them away in a lagoon somewhere or another, and as it were, saving them for later and having frequent contact with them. I mean, you'd have to look into what the mechanisms, the possible mechanisms of transmission is. But it seems to me you can tell a story about this.
Yes.
It's just that I think you need some kind of story like that.
Yes, and somebody who knows more about seals than I do. But one of the phenomenal things that's coming out now about MERS, the Middle Eastern respiratory virus, is all of this focus on camels and the ways people interact with camels. And I mean, it's astounding the photos that you see people kissing their camels, and I mean, really, the very quite intimate relationships. So nothing's off the table as far as I'm concerned. But then that's also the amazing thing about the genetics is the genetics can create these lines. And what genetics has done also in HIV, it's forced us to tell a lot of stories that we didn't really want to tell.
Yes, Mary.
Hi. I'm thinking about seals here. One of the stories I tell my history of medicine students is the transference of zoonoses to human beings. But one of the things that seems to me, and it goes back to Philip's question, it's very, very unclear exactly how this transmission between humans and animals happens. I mean, first thing my students say, well, did they eat the seals? Did they eat the chimpanzees? And we're not even going to go any further than that. But, I mean, SARS teaches us that it's a very, very complicated relationship. And is there a lot of work on the very peculiar ways and the very multiple ways in which humans and animals interact? It seems to me there must be, because that interaction is not just eating them or having sex with them or anything like that, but it's much more complicated. And even when you look at, when you develop irrigated agriculture and you end up with the transmission of many kinds of animal disease to humans, what's fascinating about what you were saying is the opposite transition. I think there's a question.
I think, as far as I'm concerned, everything's on the table. One of the peculiarities about Ebola is that they've been looking for ages for what the animal reservoir is. And the big suspicion has been bats. But every time they've been testing bats, they're finding antibodies, but not an active infection. Now, one of the questions that I have raised is, are they looking in the wrong parts of the bats? Are they looking in their testes? Are they looking in their eyeballs? Are they looking in so this whole notion of protected zones where viruses might be persisting is a question that is entirely being opened up now because of the Ebola outbreak. The other question that's been put on the table is, and it has been raised with the question of the transmission to seals as well, is do we need to be looking at for intermediary species, whether those be arthropod vectors or other kinds of small mammals or, who knows what else. But again, we have a line now to be tracing and just need to be more creative. Frankly, in putting that together.
TB is clearly aerosol transmitted, and no blood and no sex. There is nothing like that. A TB patient is normally very ill. I don't know how the people 2,000 years ago felt of TB. So I like the idea. It's interesting, but it needs still something that's missing about, in a year, a TB patient infects 15 people, more or less, on an average. So the question really would be, would a seal be more sensitive, like Ebola is transmitted from animals who don't care, as you just mentioned, who don't suffer, but they can transmit it, and humans are more susceptible than to Ebola. So the question really would be, is a seal highly susceptible to an MT, Mycobacterium tuberculosis? Because that could explain it. Otherwise, I would have, with due respect, would have a difficulty to believe if it is like human-to-human transmission. It's a very rare event.
Well,
It could be, yes.
Mycobacterium pinnipedi is a documented disease in seals.
I know that all.
So,
I know
so
really,
there's first of all, there's a question of how do seals transmit it between themselves? I don't know.
No, how susceptible would a seal be to a M. tuberculosis progenitor of the seal's pathogen? If it would be highly susceptible, that would be an interesting question. Another,
Yeah, I have no idea how to, it's simply that the genetics has already established the genetic connection between what is now modern M. africanum and modern tuberculosis in seals.
One point I would like to bring up, which always fascinates me, is in India, as you know, cattle are kind of sacred. So there are valleys in India where old cattle are brought to, and they live there, and they get reinfection with M. tuberculosis, not M. bovis. So that would be an interesting situation where you can study transmission from human beings into cattle nowadays, which would kind of mimic the situation thousands of years ago, and might be of interest to you. And because these cattle are more or less kept under wonderful kind of a heaven for them, and people there are often infected with MTB, and it's not really studied intensively, but that's a situation where humans transmit it again into
Yeah.
animals today.
Yeah, thank you.
Yeah.
Next person I have on my list is Patricia Kitcher. Maybe explain this, and I just missed it on the way.
Maybe you explained this and I just missed it on the way by. One of the points you were making was that the strains of tuberculosis that are around now are a lot worse. And I didn't understand exactly why that was. You seem to be suggesting that the lousier strains of tuberculosis beat out the less bad ones, but I always thought that it was good if you were a pathogen not to kill people too quickly because then you stayed alive. So I was just out there, and then there was a suggestion that some of these people had 2 or 3 strains of tuberculosis in them, and I wondered whether, so I was just trying to figure out the mechanism. Is it that if you've got 2 or 3 kinds of tuberculosis, you're so weakened that what you would be fighting one with is, and so it's going to throw, I just literally, one of your messages I thought was, we've now got a worse disease than we had before, and I just wondered if there was, if I'd missed the account of how.
Okay, so there's 2 questions there. Number one, the Hungarian mummies, that study found several different strains, substrains of the European lineage. So they weren't from all over. They were from the lineage we've already identified as being the typical European lineage. So you know, it keeps evolving in various ways, and so they are simply substrains. So that's the co-infection that they're talking about there. Oh, this woman died at age 28. I think the others were also, in relative terms, quite young. Well, and you saw the spine. This is someone that is a very, very serious infection, and probably a long-term infection, to do that kind of damage to a spine that was clearly while she was still a child. That was happening in the formation as she was growing up. I don't know what kinds of studies have been done about that kind of co-infection of kind of closely related types of tuberculosis. In terms of the more virulent strains, there is, in general, of the geneticists that I've talked to and heard present, there's in general a move away from an assumption that there is a universal agenda for microbes, is that they don't want to kill you too quickly because they want to keep you alive. It really depends on how quickly they think they can get, but assuming some kind of Darwinian imperative, that how quickly they can get into a new host. And one of the things that we're seeing in this modern period, which is apparently when the Beijing strain and the European strain are most vigorously developing, is that they've got millions of people to infect. I mean, that this is why I was bringing in all too quickly, but the massive migrations and the just translocation of so many people. So I don't know. I think it will be wonderful. It will be necessary to go back and look at the kinds of statistics that have been collected for tuberculosis in the 19th century, and because the general percentage is, in talking about tuberculosis, is that for every 10 people infected, only 1 will develop an active disease. So for most people, it comes into their body and their immune system contains it. But that 1 in 10, because of pregnancy, because of a co-infection, because of some other kind of infirmity, that disease will become active again. And when it becomes active, then they can transmit to another person. But has that always been the same ratio? I mean, it's actually similar to the question earlier. Are some forms of the disease more likely to instigate an active form, and therefore be more easily transmissible and perhaps more quickly lethal? Huge amount of work to be done. But one of the things that I consider amazing, that there's also work that's been done on human genetic responses to tuberculosis. But now that we've narrowed the time frame, if we have in fact narrow the time frame of tuberculosis. 6,000 years is not a whole lot of time for there to be widespread human genetic response to tuberculosis. So there's quite a bit of work that needs to be done. And the thing that I am excited about is that we can stop pretending that history is separate from the present day. Is that whether we're working with modern samples, whether we're working with historical samples, I think we've reached a point now where we can say, not that we mush them together, I think we've never had it, has it been more important to be really precise and rigorous about how we're dating, why we're dating the way that we are, how we're creating the geographic frameworks that we are. But there's a huge amount of information that we can get out of any kind of study, whether it's from historical documentation, genetic studies, et cetera.
Just a quick question about the role of non-human accidents in the transmission of disease. So you kind of made a joke about people kissing their camels, but it seems like there might be other ways in which the diseases and the pathogens are then transferred between animals and humans through objects and so non-human accidents such as contaminated water, something like that. Can you expand a little bit in that?
Yes, I did not do cholera today. It is one of my diseases. The diseases I've chosen for this project, I'm calling them paradigmatic diseases simply because I want to have at least two different disciplines that can be brought to bear on how I'm constructing a historical narrative about them. So I don't do flu. I don't know how old flu is. Some people say it's a couple hundred years old. Some people want to say it's 500 years old. I don't have a good way of establishing flu's history because there's no ADNA to document it yet. Because historical sources, even if they're describing flu, how do I know they're describing flu? They're describing aches, they're describing pains, vomiting, whatever. How many other different diseases could that be? So I've deliberately kept my focus to the kinds of things where I think I can get more or less good data. Often it's less, but I still try to work with it. All the other possibilities for transmission will relate in some way or another to other diseases. Right now, cholera is the only waterborne disease that I'm working with. It's also the only one that's out in an environment, as opposed to either these are what we call obligate pathogens that are going directly from human to human, or they are, malaria is kind of a semi-obligate, or other ones are zoonotic. I mean, kind of repeatedly zoonotic transmission. So I didn't talk about my favorite, which is plague, tonight. But again, the thing is to stress that it has to be some environment where whatever this organism is, it will stay alive so that it can be passed to a new human host. And that's always the critical thing.
I know nothing about this topic, so this is all very, very new to me. But I just wondered about the analytical meaning of the word global in the title of the talk. Is that to make a claim about a particular global approach to the history of health that is somehow different to a national or regional approach to the history of health? Or could we just do without that word? Would the substance of your research be exactly the same if it would just be called the history of health? And then what are exactly analytically the claims about the relationship between history and health? I mean, what do you want to bring into that debate? I mean, how is it going to help us understand something by going back 35,000 years or 6,000 years? Well, what exactly is it? I just need some clarification what exactly it is that we're going to understand better because of it.
Okay, so two questions. Why global and why history?
Yeah.
I started this work, started this thinking, started the questioning process almost 20 years ago because I was reading about global health, a field that has come into existence and exists in various places in public health schools and the WHO and some medical schools now. So global health came to me as that package, in some sense, already made. What I realized, though, is that most people who use the term global health don't really know why they're calling it global health, or they're using it as in some kind of euphemistic way to mean the health of people in the third world, or economic differences, or neglected tropical diseases. It is used euphemistically. I've given a lot of thought to how I'm using it, though, and the principal ways that I feel I can justify it is, number one, the diseases I'm working on are globally distributed. They are all globally distributed. They have gotten to every one of the inhabited continents. I don't deal with Antarctica. Even if the seals are down there, I don't deal with it. So global distribution is the big thing that I'm working with. Global in a sense, metaphorical sense also, that as humans, we are all subject to these diseases. And I think there is an amazing way in which a field of global history is developing that is global in the sense of looking at humankind. And that's what these diseases are. If you're human and in the wrong place at the wrong time, then you are potentially subject to these diseases. I think I am also gearing some of the arguments that I'm making toward a global health audience in terms of the ways in which I think this gets to your point, your question about history, is I don't think there is a point at which we can say absolutely that doesn't happen anymore. That is a past that we will never revisit again. I think we have revisited all of it again in the past 2 years with Ebola, of the way in which something can come out of, and again, we knew about Ebola for 40 years. Nobody thought, I mean, and this is nobody, meaning really dozens and dozens of people in the health professions who have stated this, of people not thinking that a disease of the jungle or rural areas could become an urban disease. But that's what all of these other diseases have become as well. That's what plague became. Plague doesn't normally, plague is very, very similar to Ebola in the sense of it is not normally a human disease. It does not normally inhabit heavily populated areas. But if you get the right mix of circumstances, boom, you have a pandemic. So that's my big argument about history, is that there is no separation. Smallpox is the only one that we have put a definitive end to. Maybe eradication will come with polio very soon, with guinea worm, but that's it. We are nowhere near close to getting rid of malaria, nowhere near close to getting rid of even leprosy. 200,000 new infections are diagnosed every year. Yeah, the past is us.
Well, on that grimly egalitarian